Hi, I'm Professor Detman, and today we're going to be discussing membrane functions and cellular organelles. Our membrane, the plasma membrane, is made up of phospholipid bilayer. Our phospholipid bilayer, remember, are two phospholipids that have the hydrophobic tails creating a center hydrophobic region, and then there are two hydrophilic heads on the outside and inside of the cell surface. This creates a barrier that allows things to be blocked from the outside of the cell and the inside of the cell from intermixing together. Our cell membrane is described as a fluid mosaic. This fluid mosaic means that it is constantly in motion. These phospholipids are moving from surface to surface around all the time. And it's a mosaic because there are proteins embedded within the membrane. These proteins, known as integral proteins, provide transport and functionality to our cell membrane. These proteins also are very diverse and give us our imaging of being part of the self. When we have our cells, other cells from foreign invaders do not carry the same types of proteins, and so we can recognize them and destroy them before they can attack our own cells. As we look at this phospholipid bilayer, you can see some other things attached to it also. We have cholesterol molecules embedded within the membrane. These molecules allow for more fluidity to occur. When temperatures increase, the fluidity decreases, and when temperatures decrease in the cell membrane, the fluid will increase. We also see peripheral proteins, some of them attached to those integral proteins, that help aid in the attachment of our cytoskeleton. The cytoskeleton is the part of the cell that gives the cell its framework, kind of like your own skeleton gives your whole body its framework. We also see markers on many of our phospholipids known as glycolipids. Glyco for sugar means that there are some sugar units attached to our phospholipids that help identify the cell. There are also glycoproteins, where we see proteins having sugar chains linked to them also. Once again, this aids in the cell-to-cell recognition. The outside of the cell, or the extracellular fluid, is a place where the cell can attach to different fibrous membranes. This matrix usually contains some collagen proteins, maybe some fibrous proteins, and different other salt compounds. The inside of the cell, known as the cytosol, is where we see all of our cellular organelles. Our cellular organelles, as we will see later on, are also composed of the same phospholipid bilayer structure. And that keeps the internal structure of the cell all cooperating together. If we look at some of these integral proteins in a little bit of detail, we can see that they vary in their functionality. Some of these integral proteins, also known as transmembrane proteins, can provide transport. Transport proteins allow for molecules to move in and out of the cell that cannot move freely through the cell membrane itself. Our cell membrane is what's said to be selectively permeable. And because it's selectively permeable, only certain molecules, like small, nonpolar molecules, can pass easily through it. Larger molecules, or charged ions, need to move through these transport proteins to get carried from the inside to outside of the cell or from outside to inside. Another type of integral proteins are enzymatic proteins. These proteins allow for reactions to occur very quickly. Enzymes are large proteins that just speed up the chemical reactions, and in turn, the proteins embedded in the membrane allow for reactions to occur inside the cell much faster. Another type of integral proteins are receptor proteins. These proteins actually receive signals from outside of the cell and send a chemical response into the cell for certain cellular activities to occur. They could be as simple as hormones or any other type of foreign invader sending an attack signal. Some other functions of our proteins are intracellular joining. Our intracellular joining proteins allow for two cells to adhere together. Many of our cells form tissues, and these tissues are all adhere together via these intracellular joinings and things like that. These proteins just allow them to attach together. Once again, our cell has to recognize other cells as being part of itself. Whenever we have foreign invaders, we see different markers. Our cells contain what we call cell-to-cell recognition proteins. That means when a liver cell comes in contact with other liver cells, it realizes that it's supposed to belong there. And that's why transport of foreign organs in a transplant surgery can cause many problems and rejections of the new organ. These cell-to-cell recognition proteins are very important and need to be matched as close as possible when an organ transplant is necessary. And then the last type of integral proteins we see allow for that extracellular matrix and cytoskeleton to attach to them. And this actually helps hold the cell in place and gives the cell its overall structure. So you can see that these integral proteins can vary greatly, and they don't necessarily all have to be found in the same cell. We can show different protein markings on different cells, and in turn, that's what gives it that mosaic appearance, meaning different pictures from different proteins being shown. Our cell membrane overall is very permeable to very small nonpolar molecules. This is a process known as diffusion. You're probably familiar with diffusion when it comes to Christmas shopping, for example. When you're walking down the hallways of the mall that are very crowded, you'd like to get to areas that are less dense. That's what diffusion is all about. Biological diffusion, it happens across a membrane, when molecules are very dense on one side of the membrane and want to go to areas where it's not as dense. Diffusion always occurs according to what's called its concentration gradient. Its gradient tends to flow downhill, so to speak, meaning moving from very high areas of concentration to very low areas of concentration. When these molecules move, they're going to diffuse according to that concentration until they reach what's known as an equilibrium. Remember, small nonpolar molecules will diffuse across a membrane passively, very easily, without any export of energy. They will move until they reach this equilibrium. And it doesn't matter if there's more than one molecule. Molecules will diffuse independently according to their concentration gradient. They will not move across a membrane if they are too large, but they will move if they can to reach that equilibrium. We can see a small video identifying these structures.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
Now that we've seen schematically how those molecules can diffuse across the membrane, what we can see now is that we show other molecules being much larger going across transport proteins. Remember, transport proteins are functional proteins or integral proteins that allow larger molecules to come across the membrane. These transport proteins can either be carrier proteins or they can be channel proteins. Either way, they help these larger molecules or even ions to move across the membrane freely without any input of energy. Both simple diffusion and this type of diffusion through the transport proteins, known as facilitated diffusion, occur without any energy. This is what we call passive transport. When molecules move from a high concentration to a low concentration without energy, that is known as passive transport. The simple diffusion of molecules across the phospholipid bilayer, or the facilitated diffusion where they require carrier proteins or channel proteins, just like a tunnel going through a mountainside. You can see how you can get from one side to the other without any difficulty. Let's watch another video describing this membrane permeability.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
Passive transport is talking about the movement of molecules across the membrane. When we see that movement of molecules, we also can talk about the movement of water. Passive transport can also be described as the movement of water according to its concentration gradient. But this is given its own specific name of osmosis. Osmosis takes these water molecules, and when the solutes are too large to cross, the water will cross to try and reach an equilibrium that way. Osmosis occurs according to a hypotonic solution and a hypertonic solution. A hypotonic solution is a solution that has less solutes and more water, whereas a hypertonic solution has more solutes and less water. Water will always travel from the less solutes to the more solute concentration. Or, to put it differently, water will move from its high concentration to its lower concentration, trying to diffuse out the concentration of those solutes. If we look at this image here, you can see how the water, if the solutes move, the water will reach equilibrium along with the solutes. But if we look at this image, we can see that when the solutes are incapable of movement across that membrane, that the water will have to diffuse to try and equate it out. When it moves across the membrane that way, we see it going from a hypotonic solution to a hypertonic solution. Water moves freely across this phospholipid membrane, even though those solutes cannot. Let's once again see another video describing this process.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypotonic environments, water rushes into the cell, and the cell swells, but is kept from breaking by the rigid wall layer. The pressure of the cell pushing against the wall makes the cell turgid, and is the desired state for most plant tissues. For instance, placing a wilted celery stalk or lettuce leaf in a hypotonic environment of pure water will often revive the leaf by inducing turgor in the plant cells. Animal cells lack rigid cell walls. When they are exposed to hypotonic environments, water rushes into the cell, and the cell swells. Eventually, if water is not removed from the cell, the pressure will exceed the tensile strength of the cell, and it will burst open, or lyse. Many single-celled protists living in freshwater environments have contractile vacuoles that pump water back out of the cell in order to maintain osmotic equilibrium and avoid lysis. Plant cells are surrounded by rigid cell walls. When plant cells are exposed to hypertonic environments, water rushes out of the cell, and the cell shrinks away from the rigid wall. These cells are dehydrated and lose most or all physiological functions while in the shriveled state. If cells are returned to isotonic or hypotonic environments, water re-enters the cell, and normal functioning may be restored. Animal cells lack rigid cell walls. When they are exposed to hypertonic environments, water rushes out of the cell, and the cell shrinks. The resulting cells are dehydrated and lose most or all physiological functions while in the shriveled state. If the cells are returned to isotonic or hypotonic environments, water re-enters the cell, and normal functioning may be restored.
As we discuss this osmosis, we can bring it into relationship with our own red blood cells. Our red blood cells have a concentration of about 1% saline solution. When we have our IV at the hospital, they give us an IV of 1% saline to where the concentration of our solutes is equal inside of our blood cells as well as outside. This condition is known as isotonic. However, if we were foolish enough to drink some of the salt water in the middle of the ocean, we would be drinking a solute solution that is more hypertonic compared to that inside of our cells. When we do that, our cells will shrivel up and collapse down, known as crenation. That's because the salt water is hypertonic to our cells inside of our blood. On the other hand, if we accidentally received an IV at the hospital of distilled water, then the water in the distilled water has no solutes, making it a hypotonic solution. That water then will rush into our cells, and once it enters our cells will swell and possibly even rupture. If our cells rupture, that is not a very good thing because then they create ghost cells, and we could lose a lot of blood volume in that fashion. Passive transport and osmosis both require no energy input. However, there is a lot of transports that do require the input of energy. This is what we call active transport. Active transport is actually taking molecules and moving them against their concentration gradient. That's like trying to drive in the westbound lane when you're going in the opposite direction. It's not very feasible. When we have active transport, we're trying to push these molecules in a direction they do not naturally want to go. And so in order to do that, we must put a lot of energy in. The sodium-potassium pump is an example of active transport. Inside most of our cells, we create this membrane potential. The interior of our cells is more negative, electronegative, than the outside of our cells, where we see a more positive environment. Sodium ions, which are positively charged, are in great numbers outside of our cells and want to flood into our cells. When they come into our cells, that'll cause a more positive environment inside, and our cells do not like that. So they must actively pump the sodium back out. When we look at the sodium-potassium pump, we can see that happening. First, we see the sodium filling up our proteins. These transport proteins have binding sites for the sodium ion. We need to get the sodium back out of our cells that is naturally flown in, and so we bind them to these proteins. Once that protein binds those sodium, we bring in energy. Remember, our energy carrier is ATP, adenosine triphosphate. It is a very metastable molecule which will donate a phosphate group to allow an action to happen. When that protein receives that phosphate group, it allows for the sodium to move to the outside of the cell. You can see that as it moves to the outside of the cell, that the shape of the protein changes, almost forbidding that sodium to bind back to it again. Once that happens, binding sites on the same protein open up for potassium ions to come back into the cell. When those potassium ions come back into the cell, the phosphate groups will fall off and the cell will open back up to the interior. At this point, it reopens the sodium ion channels and turn to allow for more sodium to bind again to be pumped back out of the cell. This process of active transport requires both the protein as well as energy to transport these molecules against their concentration gradient. Now, once again, let's see a video actually describing this process and showing how those proteins move the sodium ions out of our cell.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Now let's compare the two different types of transport of molecules that are too large to even go through a transport protein. Many of these are bacterial cells or even fluids that just cannot come through the phospholipid or through those channel proteins. These two processes are known as endocytosis and exocytosis. Endocytosis is the process of exporting or importing molecules into the cell. When things are too large to come through the membrane itself or through transport proteins, but the cell still requires them, they go through this process of endocytosis. This is when the membrane will invaginate down, engulfing that object or that molecule that is too large. Sometimes it will also bring in fluids, and sometimes it brings in very specific molecules. These different processes, of course, have different names. Phagocytosis is the cell eating something similar to what our bacterial cells get engulfed by our white blood cells. When infected with bacteria, our white blood cells will phagocytize them, engulf them, and destroy them once they are inside of our cell. Penocytosis is when a cell is known to be cell drinking, pulling in fluid from the extracellular matrix. And receptor-mediated endocytosis actually has markers or receptors on the cell membrane that are very specific to certain molecules and will only bring in those molecules along with a little bit of the extracellular fluid. And this is whenever the cell itself requires some specific molecules. Exocytosis is the process of exporting the cell through exporting molecules from the cell to the outside of the cell. It is a basic process that happens all the time when we're transporting proteins and other macromolecules out of our cell. Now let's see a video that actually demonstrates the invagination of our cell membrane through endocytosis and exocytosis.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
Now let's review passive transport and active transport. Remember, passive transport does not require any energy. It occurs naturally, moving molecules from a high concentration to a low concentration. Simple diffusion is the movement of molecules across the membrane itself. Facilitated diffusion is the movement of molecules through a transport protein, either a channel protein or a carrier protein. Osmosis is the diffusion of water molecules across the membrane. Osmosis, remember, occurs when the solutes are too large to move across the membrane itself. And then filtration. Filtration is one that we did not discuss, but it is also the movement of filtrates across the membrane through a passive process. To review active transport, remember, requires the protein and also requires ATP. We can transport solutes across the membrane through this process. Exocytosis and endocytosis also require energy to allow this reaction to happen. Cellular organelles. Now we're going to discuss the organelles found within our cell membrane. These organelles will resemble quite often that phospholipid bilayer and the functionality of our phospholipids. When we look at a generalized structure of the cell, we can see our eye drawn straight to the nucleus in the center of the cell. The nucleus is the largest organelle that the cell contains. It has several components inside of it, the most being our DNA. Continuous with our nuclear envelope, we see the endoplasmic reticulum. The endoplasmic reticulum has two structures, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. These two structures are connected to the Golgi vessels indirectly. The Golgi apparatus is actually where our proteins complete their processing. Other organelles found inside the cell are our mitochondria. Our mitochondria, we'll see, are the powerhouse of the cell. This is where our energy is actually produced. We also find lysosomes inside of our cells. Lysosomes act as an individual cell's digestive system. Just as we have our stomach and intestines to complete our digestive system, every one of our cells contains a lysosomes that completes that process. We also see many different structural proteins and filaments that compose our cytoskeleton. Our cytoskeleton gives our cell its framework. This helps support the cell and allows it to maintain its shape. Now this is a typical cell. Some cells in our bodies do not contain this and other cells do, and that's what we call our microvilli. You can see the microvilli down on the lower left of the screen. The microvilli are projections of our cell membrane, and these projections allow for increased surface area. Our small intestine cells have many microvilli to help in the absorption of all of our food sources. There are other organelles inside of our cell also, such as the ribosomes, the centrosomes, and the peroxisomes. All of these participate in the well-functioning of our cell. If we compare a typical animal cell to a typical plant cell, we see a lot of the same general features, such as the nucleus, the endoplasmic reticuli, the Golgi apparatus. We also see mitochondria and peroxisomes. But there are some key differences between plants and animals. One major difference is that the plant cell is surrounded by a cell wall. This cell wall is composed of cellulose and allows the plant to be more rigid. Plant cells also contain a very large central vacuole. This central vacuole is a place where we will find lots of fluids and metabolic waste products that the plant produces. And the third key difference between plants and animal cells is that plant cells contain another organelle known as chloroplast. The chloroplast is where the plants manufacture their food. This is where photosynthesis is occurring inside of our cells, or plant cells. So let's look at these organelles in detail. The first one we're going to focus our attention on is the nucleus. The nucleus is a very large structure. It is composed of a phospholipid bilayer, and it contains our DNA. This nucleus is the key to our cell functioning. It is the brain of our cells. Looking at the nucleus again, you can see a dark staining region in the center of the nucleus. This is known as the nucleolus. The nucleolus is where our ribosomal subunits will be manufactured. Our ribosomes are a non-membrane bound organelle that are a key role in protein synthesis. We create two subunits here in this nucleolus. In cells that are very efficient in protein synthesis, you may have several to many nucleoli present in those cells. Inside this nucleus, we find this double phospholipid bilayer. Now, when I say that it is a double phospholipid bilayer, that means that the phospholipid membrane actually folds back upon itself. This folding creates two phospholipid membranes. As it folds across itself all over the cell, it creates nuclear pores. These nuclear pores are the way that molecules are transported in and out of our nucleus. There are no transyl proteins inside this membrane. That's why these pores are so important. For proteins to get inside the nucleus and for RNA to leave the nucleus, we must have these pores available. Another component of our nucleus is the chromatin. This is our DNA. The chromatin is a long chain of nucleic acids. This chromatin is thread-like, hence the string-like appearance inside of our nucleus. When the DNA gets ready to divide and the cell becomes ready to divide, we see this DNA condensing down into a form called chromosomes. These chromosomes condense up nice and tight to where you can actually visualize them underneath even a light microscope. If you think about fishing line, for example, the chromatin, our DNA, compares to that fishing line that's out in the middle of the lake. You can't see it very well, but you know it's there. When the cell gets ready to divide, that chromatin condenses down in the form of chromosome. That's just like pulling in our fishing line along the reel and making it condense to where now it is very visible. Kind of gives you a good visualization of what's going on. As we look at these ribosomes, you can see that there is a large subunit and a small subunit. These are manufactured in the nucleolus and will leave the nucleus through the nuclear pores to be transported into the cytosol. The ribosomes are our key site for protein synthesis, and because of this, we can find them everywhere inside the cytosol. There are two main types of ribosomes. There are free ribosomes, and there are what we call bound ribosomes. Free ribosomes are floating around in the cytosol, producing proteins that are going to function within the cell itself. This is very important for all of our cellular activity that takes place, for the need of these proteins are an important role. We also have ribosomes that are bound. These ribosomes are attached to the endoplasmic reticulum. As they attach to the ER, as it is known as, they produce proteins that are going to be secreted from the cell. These bound ribosomes are a key player in that secretion. As we look at the endoplasmic reticulum, we can see that it is a continuation of the membrane of the nucleus. That outer phospholipid bilayer of the nucleus creates the membrane of the endoplasmic reticulum. There are two components to our endoplasmic reticulum, the rough ER that has ribosomes bound to it, and what we call the smooth ER. The smooth ER does not have the ribosomes and it appears with this softened appearance. The membrane of our endoplasmic reticulum is called the cisternae. The cisternae is where these membrane proteins are produced. As we look at this figure here, we can see that the ribosomes that are going to be bound to our endoplasmic reticulum find certain channels attached to that cisternae, and produce their proteins. As the proteins are being produced, they empty into the packaging or the space of the endoplasmic reticulum. This is known as the cisternal space. It is in the cisternal space that these proteins will continue on with their structural formation, going from secondary levels to tertiary levels of the proteins, becoming more functional. It is also in the cisternal space where they may combine with glycoproteins and become glycoproteins, where they're mixing with sugars. They can also be packaged together with lipids. These carbohydrates and lipids are produced in the smooth endoplasmic reticulum. The smooth endoplasmic reticulum does not have ribosomes attached to it and is not involved in protein synthesis. The smooth endoplasmic reticulum is actually involved in the synthesis of lipids. The smooth endoplasmic reticulum also metabolizes carbohydrates. The carbohydrates can either be created, synthesized, or they can be broken down and utilized for cellular energy. The smooth endoplasmic reticulum also is involved in storing calcium ions. This is very important, for example, in our muscle cells because they require calcium to contract, and so the calcium is stored here in the smooth ER. And a fourth function of our smooth endoplasmic reticulum is that it helps to detoxify poisons. These poisons, such as drugs and alcohol, can get into our system and need to be broken down. We tend to see a lot of smooth endoplasmic reticulum in our liver cells because of the harshness of the chemicals that we ingest into our body. All of our medications, from aspirin to prescription drugs, are manufactured and processed through the liver cells and the smooth endoplasmic reticulum. That's why many side effects could be liver damage. So to recap what the smooth ER does is it synthesizes lipids, it metabolizes carbohydrates, it stores calcium ions, and it detoxifies our poisons. The rough ER, on the other hand, is where these secretory proteins are being manufactured and put into their tertiary configuration. So if we move back over to the rough ER again, we can see that these proteins, once they are combined with lipids and carbohydrates from the smooth ER, will then be packaged into a little vesicle. This is the budding off of our phospholipid bilayer into a little package or a little mini cell. This vesicle will be carried to our next organelle, known as the Golgi apparatus. The Golgi apparatus is the key receiver and manufacturer of these proteins, the one that puts it all together. The Golgi apparatus also has a phospholipid bilayer that's known as a cisternae. It also has a cisternal space on the inside. But a way to think of the Golgi is that they are separate membranes, almost like a stack of pita breads. You know how pita breads have the pockets inside of them and the bread on the outside? That's a lot of how our Golgi apparatus are arranged. Our Golgis have also a very strong polarity. The receiving side of our Golgi apparatus is known as the cis face. It will receive the vesicles from the endoplasmic reticulum. Once it receives these vesicles, it will distribute those vesicles into the cisternal space of the Golgi. Here, the Golgi will actually process through those proteins that have come from the ER, those lipids that have come from the ER, and those carbohydrates. It will repackage and sort through them and ship them out from what we call the trans face. The trans face of our Golgi apparatus is the shipping side of our Golgi. When we see this happening, it can actually fuse with the next membrane down on the cis side again, resort through them, repackage these proteins, and ship them from the trans surface again. So this process takes place until those proteins have reached their overall conformational form. I like to call the Golgi apparatus Mr. UPS because when we think about what UPS does, is that they receive packages, they sort through those packages, they repackage those packages, and they ship them back out. And that's exactly what our Golgi apparatus is doing. It's making these proteins functional to be removed from the cell or to go and do their work in the cell in the form of other organelles. One organelle that is packaged in the Golgi apparatus and leaves as a vesicle is what we call our lysosomes. Lysosomes are the cell's digestive organelles. Lysosomes contain what we call hydrolytic enzymes. These hydrolytic enzymes will break down all of our major food components, such as nucleotides, carbohydrates, proteins, and even fats. What happens is that the cell will bring into itself, through endocytosis, large molecules, such as bacteria. These large molecules will then fuse with the lysosomes. Remember, lysosomes are also a phospholipid bilayer, and so the membranes will fuse together nicely. Once they fuse together, these hydrolytic enzymes are released and help to digest those large particles. Lysosomes also, though, help maintain the compartments inside the cell. If there are dead organelles or organelles that are no longer functioning properly, the lysosomes will fuse with them also and help digest those molecules. This is what we call autophagy. Let's watch a video about how these lysosomes actually perform these functions.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
Another component that is budded off or created through the Golgi is what we call the central vacuole, or vacuoles. The central vacuole is significant to plant cells. Animal cells do not have them. Animal cells do, however, have vacuoles known as contractile vacuoles and create food vacuoles. These vacuoles, however, are also part of the components coming from the Golgi apparatus. The central vacuole in plants is a place where large amounts of water can be stored. As the plant grows, these central vacuoles can take up almost 90% of the interior of the cell, pushing all of the organelles out towards the outside. But it's not just water that's found inside these central vacuoles. These central vacuoles contain metabolic waste products that the plant cell doesn't really easily get rid of. Think about some of your metabolic waste products. Those are released simply through sweating or through our urinary system, things like that. Plant cells cannot go through these things, and so they contain those metabolic wastes in their central vacuoles. Plant cells also contain ions, calcium, potassium, chlorides, things like that in the central vacuole. They contain pigments. The flower colors that you see on the plants contain pigments in those central vacuoles. They can also contain poisons. Many plants are poisonous to different insects or birds or berries even. Those metabolic poisons are also stored in the central vacuole. So you can see that it has a lot of diversity to it. The organelles that we have just discussed are all part of what's known as the endomembrane system. The endomembrane system is a system within the plasma membrane, the group of organelles that cooperate together in the form of the same type of phospholipid bilayer. But an underlying reason of this endomembrane system is for the production and secretion of our proteins. The endomembrane system begins with the rough ER. The rough ER is a continuation of the nucleus. The nucleus, remember, has a double phospholipid bilayer. The outer membrane of the nucleus creates that rough ER. The smooth ER also is continuous with the rough ER. This is the beginning of our endomembrane system. The endomembrane system is a group of organelles that are connected directly or indirectly by this phospholipid bilayer. The ER and the nucleus are connected directly. When those proteins are produced, however, they bud off in a vesicle, and that vesicle carries those components to the Golgi apparatus, which is our next part, organelle of the endomembrane system. Remember, it is in the Golgi apparatus that we see this processing take place, this repackaging of those proteins, the functionality of all of these molecules occurring. The Golgi apparatus will then bud off vesicles. Those vesicles can go to form lysosomes and vacuoles, or they can actually be exported from the cell through exocytosis at the plasma membrane. When we look at this overall endomembrane system, we can see this continuation, the flow of those proteins throughout the system, all the way through to the budding off of the plasma membrane or the secretion of the lysosomes. Let's watch a dynamic video showing this expression.
A secretory protein is synthesized inside the rough endoplasmic reticulum, migrates through it, and exits inside a vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein. The protein is dispatched from the Golgi in a transport vesicle that then fuses with the plasma membrane, releasing the protein to the outside of the cell.
Now, two more organelles we need to talk about. These two organelles are not part of the endomembrane system. They are made up of phospholipid bilayers, but they have nothing to do with the secretion and synthesis of our proteins. These are the mitochondria and the chloroplast. The mitochondrion is the powerhouse of the cell. It is within the mitochondrion that we produce all of our cellular energy. The mitochondrion is capable of taking in plant products such as glucose or other products like proteins and lipids and breaking them down into very simple inorganic components. In the process of releasing all of the energy from these molecules, we in turn produce energy, the form of ATP. Let's look at the mitochondria itself. It is made up of two membranes, an outer membrane and an inner membrane, almost looking like a kidney bean, doesn't it? The outer membrane is the surrounding area of the mitochondrion. Between the outer membrane and the inner membrane, there is a space known as the intermembrane space, or between the membranes. The inner membrane is a little bit more detailed. It is this inner membrane that starts to take on lots of folding. These folds are known as cristae. Cristae helps to increase the surface area, and it is along these cristae that the bulk of cellular respiration and the most of our energy is actually produced. Inside the cristae is another space. This inner membrane space is what we term the matrix. The matrix of the mitochondrion is very unique. It is one organelle that shares similarities to our nucleus. The reason being, the matrix contains DNA. It's known as mitochondrial DNA, and it is very unique because the mitochondria then are capable of reproducing themselves without relying on the nucleus. This mitochondrial DNA can be tracked through the maternal lineage of anybody because it does not mutate it always remains the same. This mitochondrial DNA allows for us to produce more mitochondria in the event that we become more active, because the mitochondria are the key to our energy. It is where we convert glucose and oxygen to carbon dioxide and water. It is where we produce all of our ATP. Because mitochondria have their own DNA, they also contain their own ribosomes. Remember, ribosomes are the key component to protein synthesis. These proteins are the enzymes that will help allow for cellular respiration to occur. These proteins are a necessity for life to exist. The mitochondria can be from anywhere from hundreds to thousands of them in your cells, depending on your activity. If your activity decreases, the mitochondria will phase out and the lysosomes will recycle them. If your activity increases, they are capable of reproducing without having to reproduce the cell itself. So we have an outer membrane and an inner membrane. The inner membrane produces many folds known as cristae, and inside that inner membrane we have the matrix. These are all components that are going to become very significant as we continue on in this course. The chloroplast is another very important organelle found in plant cells. Animal cells do not have chloroplast. Plant cells, however, have chloroplast in only some specific cells, because it is the chloroplast that allows for photosynthesis to occur. The chloroplasts actually bring in carbon dioxide and energy from the sun and can produce sugars, and they release oxygen, which is a necessity to us, as a byproduct. These chloroplasts are arranged different than the mitochondrion. The chloroplasts have an outer membrane, and they have an inner membrane with an inner membrane space, which is very similar. However, their functional units occur within that intermembrane. They have inside there little discs known as thylakoids. These thylakoids are where the light reactions of photosynthesis occur. The thylakoids, too, are made up of phospholipid bilayers and have a thylakoid space inside. The thylakoids are arranged as stacks inside the chloroplast. These stacks are known as a granum, one stack. And if you are surrounding these stacks of thylakoids, or these grana, is a semi-fluid medium. This fluid is what's termed the stroma. The stroma is where the dark reactions of photosynthesis occurs. Inside the stroma and inside the thylakoid, we have the conversion of carbon dioxides into starches. We get all of our food directly or indirectly from our plants because they are the ones that can manufacture it. They can go through autotrophic production, which is self-feeding. We must consume the plants or animals that have consumed the plants in order to receive any of our energy. We are not capable of producing our own food. So you can see that the chloroplasts are as extremely important to plants as they are to us. Something else about the chloroplast that they share with the mitochondrion is that they too contain their own DNA. They too contain their own ribosomes, and so they are capable of their own production to keep those enzymes prepared and fresh inside the chloroplast in order to do all of the work that they must do. So to recap, our mitochondria and our chloroplasts both contain DNA. They both contain ribosomes. Plants and animals both have mitochondria, but plant cells only have the chloroplast. And it's only the photosynthetic plant cells themselves that contain the chloroplast.